This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 873, recorded on March 1st, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. And at Westover Air Force Base up the road, it's 37 Fahrenheit, 3 Celsius, and overcast. We're supposed to get some rain later today.
Also joining us from Austin, Texas, Rich Condit.
Hello, everybody. We got 71 degrees, and up until just a few minutes ago, it was sunny. The clouds are moving in, but it's a very pretty day.
And from Madison, New Jersey, Breanne Barker.
Hi, great to see everyone. It is 45 degrees Fahrenheit, 7 Celsius here, and rather cloudy. My weather app says that it is raining, but my eyes disagree.
Trust the app. Definitely trust the app.
And your weather is I'm in New Jersey today, so your weather is my weather, even though I'm in the basement and I don't have my phone. So I'll take your word for it. You're 13 miles away, so it's probably similar. We have one PSA from Abercams, the annual biomedical research conference for minority students. They're launching a new meeting, the Abercams ePoster Spring Symposium for Emerging Scientists, for selected high school students who've conducted research, community college students, and first-year undergrads to present research in one of the 12 Abercams disciplines. The meeting, held online Saturday, April 23rd, 2022, will help students prepare and qualify for subsequent Abercams participation in the annual 4 meeting. Abstracts are due March 15th. In addition, Abercams wants to promote the professional development of early career scientists by encouraging grad students and postdocs to sign up to be abstract reviewers and judges. Abercams 2022 will take place November 9th through 12th in Anaheim, California. Additional details will be available in the spring. All right, so today we have a snippet and a paper for you. And for the snippet, I thought we would talk about porcine coronaviruses, because it turns out there are some very interesting lessons to be learned. And we don't talk enough about non-human animal viruses. And so I picked a review article.
Especially these days.
Yes, indeed. I picked a review article called Porcine Coronaviruses, Overview of the State of the Art by Hanna Turlowitz-Podbielska and Malgorzata Pomorska-Mol from the Poznan University of Life Sciences, which is in Poznan, Poland. They have a lot of pigs in Poland, so they are interested in this issue. This was published March of 2021 in the journal Virology Sinica, which is published by the Wuhan Institute of Virology. I have to say, there are lessons here for coronaviruses, but also for enteroviruses, which I will point out to you when we get to it. I sent this to Amy today. She said, wow, I guess we should be paying more attention to it.
Somebody should put some grant money into that.
All right. So remember,
and this is, by the way, this is open access, and it is a very thorough review. So if you are interested in this subject, it's a great paper to download.
Please be interested, because there are
we're going to
lessons
try and make you
learned.
interested in this
subject. We'll make you interested. So there are 4 genera of coronaviruses, as everyone should know by now. Alpha, beta, gamma, and delta has nothing to do with the variants. Okay, right. They're different. They were here before the variants.
We're just not very creative with our naming. We go for Greek letters pretty often.
Yes.
And there are 6 different coronaviruses that we know of that infect pigs. And 4 belong to alpha coronavirus, 1 to beta coronavirus, 1 to delta coronavirus. And we're going to talk about these today. They have acronyms that we're going to use. So here we go. Transmissible gastroenteritis coronavirus, TWiV. porcine respiratory coronavirus, PRCV, porcine epidemic diarrhea virus, PEDV, swine acute diarrhea syndrome coronavirus, SADS coronavirus, best name of all, right? And hemagglutinating encephalomyelitis virus, PHEV, and then porcine delta coronavirus.
And
all right, so
all except the last
people,
one have very descriptive names in terms of pathology.
Yeah,
Yes.
I was hoping that people had noticed exactly that, right now, many people are thinking about coronaviruses and think about respiratory, but in those names,
Yes.
we heard about gastroenteritis, but also respiratory. We heard about diarrhea virus, diarrhea syndrome, encephalomyelitis virus. So this already perhaps might pique someone's interest about coronaviruses.
Yeah, there is a bit of a diarrheal theme in these porcine viruses, which sounds, I can imagine from the pig farmer's perspective, this must be a really awful experience, and from the pig's perspective, too.
I have to say, we did cover SADS on TWiV
We did,
some time ago,
yes.
before the pandemic, right? It was a very interesting paper describing the outbreak in China. Now, for among these 6 coronas infecting pigs, and we should say that's what we know of, there are likely to be more.
Yes.
I mean, and these are pigs that we raise domestically. There are feral pigs, and I'm not sure that anyone has looked in feral pigs for viruses. I'm sure.
Well,
they have
there's an interesting thing with pigs. The feral pigs and the domestic pigs are the same animal,
right?
And if a domestic pig gets out, it becomes a feral pig immediately. They're like cats. They're they just immediately go wild. So the swine in the U.S. are all imported. They were brought over by Europeans, and if you are in, say, Texas, you probably hear a good bit about feral pigs, feral swine. They're a big problem. They root up stuff. The ones that are already out are out there reproducing in vast numbers. They destroy crops. They destroy property. They displace native species, and they're dangerous. They're these are big animals that, when they're not domesticated, can be very hazardous to your health to encounter. And so those are out there, and they have frequent contact back and forth with the domestic pigs, which makes it very, very hard to contain any kind of an outbreak of viruses like this.
So the feral pigs in the U.S. have been here for many years.
Many hundreds of years. Their population has increased in recent years and spread, ironically, in part because of hunting. Hunting pigs is very popular. I mean, they're made of bacon, so that's and they're an invasive species. What's not to like, right? So states that have this problem have encouraged people to go out and hunt them, but that's led to a boom in the popularity of pig hunting. And that, in turn, has led some people to transport pigs and release them in other states. And they have spread to places that they are very unlikely to have gotten to on their own. And that's likely due to hunting. And then they establish these breeding populations.
We're doomed.
Yeah.
I many years ago, in 2015, I think, I heard a wonderful talk at ASV by Amy Vincent. And it was all about swine and swine influenza, and she knew all about swine trafficking around the world. So I wanted to get her on. I mean, her name is like a portmanteau of 2 TWiV hosts already. Right? But she's at the Department of Agriculture, so she had to ask for permission. So I have to get back to her. I'd love to have her on to talk about it.
That'd be great.
But so, Alan, a pig, when it escapes, it's competitive. It can live out there. It can find food.
They can survive in the wild just fine. And they'll find they root for their food very destructively. And in many places where pigs are raised in large numbers, if they get out, they'll probably find a local sounder of wild pigs and get up with them immediately.
Interesting.
Yeah.
Pigs are smart, aren't they?
They are. And they are also, of course, transported commercially around the world by pig operations and around localities. And that's something that comes up in this paper because when they're transported from one farm to another, they can carry with them whatever they had on the previous farm.
So, Alan, why do you know so much about pigs?
I got interested in this a while ago for something I was covering, and then I kind of dug into it. I was like, wait, how do we have an invasive species made of bacon that we cannot control? That didn't make any sense to me. That, like, shouldn't this just be extirpated from the wild? You go shoot them, eat them. And then as I looked into the biology of them, it just kind of drilled down into a rabbit hole. I was like, wow, these are an incredible invasive species. They reproduce extremely well. Oh. And they're people have done analyses where it turns out you'd have to kill something like 60% of the local population every year, year after year, in order to actually reduce them.
Wow. How many are in the U.S.? Millions?
Many, many, many millions.
I've never seen one in the wild. They're probably good at
hiding.
They're
not up in the northern states. We don't really have big populations of them.
So in the farms, they're pink mostly, right?
Right.
Or maybe blotched. When they go in the wild, do they darken up so they can hide better?
I think the reason for that may just be that they live longer.
Oh, I see.
Commercially raised pigs are born and full-sized in very short order, and then they're meat.
How long does a wild pig last?
Probably a few years.
Interesting. I find this very interesting.
Yeah, it's that's how I got to digging into it. I was like, wow, wait a second. This is a complex subject.
Okay. Anyway, back to the viruses. Three of these viruses, TGEV, PRCV, and PHEV, have been in pigs for decades, apparently, and 3, PEDV, the Delta, and SADS, apparently are newly emerging, and we'll talk about that. Now, in addition, in Italy, there's a recombinant between TGEV and PEDV. It is called chimeric swine enteric coronavirus, SECOV, SECOV, and apparently it has only been found in Italy so far. Now, none of these appear to infect people, as far as we know. And of course, it's one of the questions is, does SARS-CoV-2 infect people? And they say probably not.
Infect pigs.
They
infect pigs, people. Yeah. Yes,
we're pretty sure it infects people.
It does, yes. SARS-CoV-2 does not appear to infect pigs. Yeah.
Which was interesting because it appears to infect pretty much everything else. And
a lot of things, yeah.
Pigs are very similar to us, and I was like, wow, that's interesting that it hasn't made the leap to pigs.
Okay, so let's talk about some of these viruses. As I said, there's some lessons to be learned. Porcine respiratory coronavirus. I love this sentence. Is common in the swine population and does not cause noticeable problems. Okay, it's a typical coronavirus. And here's the cool part. It is a naturally occurring deletion. The virus has a deletion in the spike gene. It was originally TGEV. Somehow a deletion occurred in the spike gene, and then it became PRCV. PRCV is a respiratory virus. TGEV is a gut virus. So this, and we'll talk more about this, but this deletion in the spike gene changed the tropism from the gut to the respiratory tract and essentially eliminated virulence, because as you'll see, this doesn't do very much to pigs.
I was going to ask, do we know if it has also changed transmission patterns?
I don't recall they say anything about that.
I don't remember.
Except that my recollection is that PRCV is pretty widespread.
It is.
Right.
And it's kind of, my take on this is that it's a variant that basically displaced TGEV.
Yeah.
That's sort of the sense I get. I must say, when I look at in the text, it sounds like the deletion of the spike protein is the only difference. If I look at the genetic maps, they're different. Or it's, looking at these little blocks and stuff, it looks like there's more going on than just S.
Right.
But they seem to think that the change, the deletion in S
It's the important one. Yeah.
is the important one.
And that was presumably what I get from that is that the spike deletion led to this change in pathogenesis and in location, and then probably other mutations accumulated now that it had diverged into effectively different species.
Yeah.
Right. And maybe we'll go into, you're going to go into the mechanisms of the change in pathogenesis.
Yeah. I think, though, this is really one of the few examples I know of a virus that has a certain tropism and it changes dramatically, gut to the respiratory tract. That's pretty dramatic. And it's also becomes less virulent. And so, we always talk about should viruses become less virulent, and I don't know examples. And this is one. And it may have been just a random deletion that occurred and it was fit, and it could transmit among pigs and infect somewhere else, which is perfectly reasonable, but obviously it's very rare.
Yeah, that was why I asked about transmission, because this would, if we were to change between respiratory transmission and fecal oral, which perhaps one could guess from a respiratory virus and a GI virus, that would be the only example that I knew of where a change like that had happened too.
That's right.
Yeah.
That's right. So this one I think is worth incorporating into the virology course.
And it may be that has occurred many, many more times than we realize.
Oh,
but
sure.
The new version has simply supplanted the old version, and now we don't have the comparison to make. But in pigs, we happen to have both viruses, and we can.
Yeah, I think you're probably right. It's happened in other situations, but we just don't look everywhere, and it hasn't happened in humans apparently. So
and the ancestor of it may be lost to time.
Yep,
may have gone the way of horsepox.
Especially since the new virus is not very pathogenic, from a mile up, the observation is that the pathogenic virus disappeared.
Right.
Period.
Yeah.
Okay. You got to be looking closely to pick this up.
So this was first picked up in 1984 in Belgium, where they were doing serological surveys, and they said all of a sudden a huge increase apparently in animals with antibodies to TGEV, but it turned out that this is cross-reactive. So it was actually this virus. And by 3 years, it had spread to 100% of the swine farms in Belgium. And subsequently, it's been found all throughout Europe, and first detected in the U.S. and Indiana in 1989. And it has, it spread quickly without causing significant health problems.
I think the pigs run a lab somewhere, and they engineered this thing and released it.
Yes.
Right?
Of course. Gain of function. And if you read the pig newspapers. Yeah. So there's cross-reactivity, right? The antibodies against one will neutralize the other. And they say this is probably how TGE has been eliminated and is no longer a significant problem for pig farmers in Europe. So that is very cool. A naturally arising deletion mutant, less virulent, different tropism, spreads throughout the population, no more TGEV.
Became its own vaccine. Yeah.
Fascinating. So TGEV is an enteric
which actually
virus.
Supports the theory that the pigs created it.
Yeah.
Of course. TGEV, enteric pathogen, reproduces in the enteric tract. It binds aminopeptidase and sialic acid as cell receptors. PRCV, that deletion in the spike eliminates the binding to sialic acid. So that virus now only binds aminopeptidase, and that is what it's binding to in the respiratory tract. But something else is going on there, not because there's APN in lots of places, aminopeptidase N. So that's not the only change responsible for that. So if you're infected with PRCV, you make antibodies, and they say it's really hard to figure out if the antibodies are against PRCV or TGEV. And if you listened to Amy a couple of weeks ago, kind of a similar story with enteroviruses, right? And so they say that we're trying to develop ELISAs to monitor TGEV versus PRCV, right? We need to know where they're circulating. They say, for example, if your herd is completely seronegative for both, then you're at risk for an outbreak of TGEV,
right, of TGEV.
which is not gone. It's just much reduced. So I thought That was very cool. Now, you can do a lot of experiments in pigs. In fact, there are laboratories that do this. And in fact, Steph Lango came from a lab in Iowa she used to wrestle 600-pound pigs. Well, no, not really, but she worked with really big and really small pigs. So you can do experiments. So they know, for example, that how long the antibody response. So they say PRCV neutralizing antibodies are gone 1 year after infection, and the pigs can be reinfected. So, but they don't get sick. So, hey, does that sound familiar?
Yes.
A little bit.
Gosh, it's just, and all of the armchair epidemiologists probably have never thought about this stuff out there. So I think that's very cool. There is a vaccine, a PRCV vaccine. I don't know why you would do that, but it's a recombinant adenovirus with the
to protect
spike
against the
TGEV.
TGEV.
Yeah, maybe that's right. But apparently it's not. It's only been tested, and it partially protects piglets from challenge, but it's not been licensed. It's not available commercially. So maintenance of a PR.
Oh, go ahead.
Maintenance of a PRCV negative facility may be achieved using strict biosecurity. And it's important because export of pigs, they don't want you to have viruses. They want you to be PCRV negative. So biosecurity means
you keep them in a brick house.
You keep them inside.
Not sticks,
and you don't,
not straw, brick.
You don't let strangers in. Many years ago, we were up in northern Canada in the west near north of Edmonton, north of Calgary. And there were big pig farms up there. And my daughter was young at the time, and she had a piglet thing, right? So we took her to see a piglet, and they said, I'm sorry, we can't let people in. We have biosecurity here.
Yeah.
I said, I don't blame them, right?
Yeah.
And they said, well, and we can't bring one out because then we can't bring it back in.
You can't bring it back in, right.
So, that's biosecurity. Yeah. Sorry, go ahead, Brianne.
I was just going to say that the fact that the pigs are protected, even though the antibodies have waned, is really interesting and makes you wonder about what other types of immune responses the pigs are making to these viruses. And the fact that the antibody responses wane after about a year is very curious. And again, makes you start to think about what is it about these viruses? If we think about things like HKU1 or OC43 and length of antibody production, I often think about how many times, in fact, when I was in high school, people used to be like, you should go cure the common cold. And I would laugh at them and be like, no one wants to cure the common cold. And I sort of like, if I had actually tried to figure out some of those answers back in the day, that would have been really useful for us right now. And the same thing, why are these antibodies only present for a year?
I think we have a lot of information because we can infect pigs in the laboratory, right? In part. Okay, so porcine hemagglutinating encephalomyelitis virus, PHEV, causes vomiting and wasting, and sometimes a polio-like encephalomyelitis, an inflammation of the brain. And they say this was the first swine coronavirus to be identified. It's the only one that's known to be neurotropic, that is, infects neural cells. The first outbreak in 1957 in Canada, and the virus isolated in 1962 using pig cells in culture and extracts of the brains of piglets that had signs of polio encephalomyelitis. And again, this one, serological surveys have shown this is quite prevalent and mostly circulates without causing disease in swine herds worldwide and all over the place, including the U.S. So it's endemic in the U.S. and it's endemic in many other places, South America as well. And apparently only pigs can be infected with PHEV. Now, here's the cool part. So this is a respiratory virus, which occasionally makes its way into the brain and causes paralysis. Does that sound familiar to anyone?
A little bit, yeah.
Enterovirus D68 in kids, right? It's doing a similar thing. causes a respiratory infection and rarely gets into the brain and causes paralysis.
And is in a
the people
virus
in
family that also has some members that cause gastroenteritis.
That's right. So the enterovirologists are trying to figure out, and by the way, they say viremia is not involved in the spread of PHEV from the respiratory tract to the brain. And it's the same thing with EVD68. In fact, there's no viremia. So how does it get there? And one of the theories is it enters nerve endings. And guess what? They know this for this virus. The virus replicates in the respiratory tract, nasal mucosa, tonsils, lungs. The virus spreads through the peripheral nervous system to the central nervous system. And they talk about all the specific nerve pathways that have been studied and that are used. Access to nerve pathways seems to play the key role in the development of clinical signs, while viremia does not. I sent this to Amy this morning. She said, we can learn a lot from veterinary virology.
Yep.
So I think that the same thing is likely happening in enterovirus D68. It's just harder to study in people, right? That's the problem. So basically, this virus is a problem in young piglets, right? Yes, less than 4 weeks of age. Anyway, this is a problem in piglets, less than 4 weeks of age. Mortality can be 100%. If you're older than 4 weeks, no problem, which I guess is partially due to immunity. And what kind of immunity would be interesting, as Brianne said earlier. And again, there's no vaccine, and so you need to do biosecurity to keep this off of your farm.
And this is one of, I don't think it's the only one of these viruses where apparently the passive immunity from the mom, from colostrum and milk, is a big deal.
That's
because
correct.
they're particularly susceptible as piglets, and if you can get past that, you're in much better shape. And there's a very small window here.
Yes.
It's just the first few days where you're particularly susceptible, and then if you can get past that, you're okay. And so if you can get maternal antibody from the mom, you get through that and you're okay.
And I think that's actually one of the reasons why Steph was has been interested in this system, is because some of the details of maternal antibody, both in terms of milk and in terms of antibodies crossing the placenta, are unique in this system.
Correct. So her advisor was Linda Safe, who's at Iowa and does a lot of work on these pig coronaviruses. So they do say maybe we should promote circulation of this virus on farms and make sure the young sows and the gilts are exposed, and then they can pass antibodies on to the piglets and protect them. Yep. All right. So now those are the respiratory coronavirus. Now we move on to the enteric coronaviruses. There are 4 of them, and they say these can be a real problem in pig production because they cause gastroenteritis. And of course, that's not good. You get diarrhea, you lose fluids. You can't put hundreds of pigs on intravenous fluids, right? So you're going to lose a lot of them. And piglets seem to be more susceptible to infection for a variety of reasons, like their stomachs are less acidic, their enterocytes renew less frequently in the lining of the intestines. So normally, ours turn over very quickly. So if a virus is killing those cells, it doesn't really matter. But in piglets, they don't turn over so quickly. So, and then they get diarrhea, they lose fluids, and so forth. And the mechanism is that the virus is destroying these enterocytes, and that causes fluid imbalances. So PEDV and SARS-CoV probably came from bats. And Delta, PD-CoV, probably originates from sparrow coronaviruses.
Yes.
Sparrow coronavirus.
That's a spillover host you hadn't thought about, huh?
No, not at all. Although the penguins in the Bronx Zoo periodically get malaria from sparrows because there's an avian malaria that infects them, and the birds are fine, but periodically they give it to the penguins and they get sick. And we know this because we did an episode of TWiV at the Bronx Zoo, and we had the director Callie, something Callie was his name, who told us all about this. Okay, that's cool. Spillover from sparrows. Yeah, they're all over the place. They're a real problem. They're
They're a
everywhere.
real problem. They're everywhere.
Does SARS-CoV-2 infect sparrows?
Gosh, do you think anyone's looked?
I don't know, but now I'm curious.
That's maybe worth looking. Yeah.
I think the virome of sparrows and other birds must be fascinating.
Yeah, it must
I
be.
think we
Sure.
limited study. Okay, transmissible gastroenteritis virus, which we have touched on, which at one time was a real problem globally until PRCV arose, right? So TGEV was first described in 1935. The virus identified 10 years later. And they say here, it's very interesting, it probably circulated earlier in pigs without significant influence on productivity or health and became important when the pig industry intensified, right? It got huge at a certain point. You probably know when it became huge, Alan, right?
I don't know exactly when, but I think it correlated with the growth of agriculture between the wars and especially post-World War II. So we went from animal farms where you had, maybe a few pigs and maybe a few cattle, and then there was specialization and more intensive farming of one particular animal.
Larger population sizes.
Larger population sizes. That's what I was going for. Yes.
Yeah. But so when this was a problem, this virus, it mainly affected young pigs because as you get older, if you survive, you've been infected, you have antibodies, and you don't get severe disease. This virus is closely related to feline and canine coronaviruses. So maybe they have a common ancestor. And they say maybe it may be able to replicate in dogs, cats, and foxes without causing disease. And maybe birds are mechanical vectors. In other words, they may spread it, but it might not replicate in them. But it's good that TGEV is no longer an issue because of PRCV. I think it's very cool. And now we have porcine delta coronaviruses, which causes diarrhea in piglets. And this one, also there's some hint that there's a common ancestor with a bird virus. So they say recent spillover between birds and mammals might have led to this. They say could be quails and sparrows. And they say this virus also infects a wide variety of cells, including porcine, human, and chicken cells. So there you go, porcine delta coronavirus. It was identified as HKU-15 in 2009 during surveillance in China. However, it wasn't known to be involved in causing disease until January of 2014. It emerged as a new swine disease-causing virus in the U.S. And so these viruses have 99% nucleotide identity to viruses previously circulating in China. I remember talking with XJ Meng about this many years ago when I visited him at Virginia Tech, and he said, yeah, it's this very similar. It may be a contamination of some kind, but we can't prove it, right?
It's the sparrows.
It's the sparrows, yes.
So it was introduced in the U.S. 2014. It has spread through most of the states, although lower, they say, than PEDV. And the infection rates are quite high in China, 24% seroprevalence, higher in suckling pigs, but they think it's probably even higher because that's just diarrhea by looking at diarrhea, animals with diarrhea.
Symptomatic ones, yeah.
Symptomatic ones. Then we have swine acute diarrhea syndrome coronavirus, which probably has been around since 2016. It caused a huge outbreak in China in 2017. 24,500 piglets died and then emerged again in January 2019 in Guangdong. Not been reported outside of China yet.
I think we talked about this back at the time one or the other of these was happening, right?
Yes, I think it was the first one. SARS-CoV
2017.
TWiV. Let's see. No, that's not it. Wake up and smell the pandemic. Nope.
No.
It's not coming up easily. Usually it comes up right away. No, it's 673. No, I'm sure we talked about it. I'm quite sure. Anyway, so this is has similarity to a coronavirus found in bats called HKU-CoV, identified in 2007, Rhinolophus bats. So maybe this one came from bats into pigs. So it's a pig pandemic virus, but it's only in China. I guess it's not a pandemic, right? No vaccines against that one. But that really blew up China's swine industry, didn't it?
Yes.
Nolan?
Yeah, they had a series of these sorts of things. So actually, I just found TWiV 546 from May of 2019, Delta Blues and Chitlins. Might be it. Yeah, so they had SADS in 2019, and there was African
swine
We
fever.
talked about African
yeah.
swine fever in Europe. I don't know that was as much of a problem in China. I seem to recall a circovirus issue in China, either that or a parvovirus issue.
Right.
It was a different virus that was
I think that's correct.
really
Yeah,
running through the pigs.
Right.
Circovirus, yeah.
The African swine fever, though, they they lost a lot of pigs to that in China, and that was January 2020.
Yeah, I kind of remember there being a problem with having a sort of issue with pig or with pork availability.
Yes,
correct.
Yeah, and both of these, African swine fever and SADS, had major impacts, and it's a huge part of the diet in China, and so it's a big deal.
Some people at the beginning of COVID said that might have been a trigger for more wildlife consumption, but I think that's so not likely because it's so expensive. Right?
It was a reasonable theory initially, but people have analyzed it, and it looks like that was not.
Yeah,
it wasn't like people made a huge switch over to more expensive wildlife when the cheap pork became more expensive.
All right, then we have porcine epidemic diarrhea virus, first in the UK, 1971. Again, causing high mortality in piglets. A virus identified in 1978. It's now considered endemic in Asia and has been reported in many European countries, but more of a concern in Asia. Substantial economic losses there. Outbreaks characterized by 80 to 100% illness and 50 to 90% mortality rate among suckling piglets. 38% positivity rate in pigs from 39 farms across 8 provinces. Came to the U.S. in 2013, spread rapidly across the U.S. So it can go from farm to farm quite readily. So they say PDV has become an important swine pathogen, ranking third after African swine fever virus and swine reproductive and respiratory syndrome, PRRSV, a virus of a different family. Similarity of strains found in China and Mexico, and the timing suggests outbreaks may be associated with the PEDV emergence in the U.S. So another puzzle where they came from. What's the origin? Seems to be closely related to a Scotophilus bat coronavirus, BTCOV, and to other known alpha coronaviruses. So maybe, again, bats to pigs. No vaccines. Again, biosecurity is needed to control this. The rest of the paper, they talk about the pathogenesis in the enteric tract, and I don't want to really talk about that, but I want to talk a little bit about transmission, that's all. They say the porcine coronas have similar transmission routes. They're spread by per os, the feces of infected pigs. It has a lot of virus in it, and of course, pigs like to contaminate themselves, so they easily eat that and
well, in the conditions
infected.
in which they're being kept, they may have more. Pigs are inherently not as filthy as the reputation would suggest. But
We make them filthy.
yeah,
right?
there's limited space, and porcine diarrhea. There's going to be a lot of diarrhea, and
Yeah.
probably aerosolization of virus. And I hope nobody's eating while listening to this episode, but they should know better with TWiV.
They say contaminated trailers and feed ingredients can serve as vehicles to transmit. And aerogenic transmission is confirmed for both PEDV and Delta coronavirus by ingestion of aerosols containing virus particles. That's interesting.
Yeah.
Okay.
Pig poop spraying everywhere.
Indeed. So I think that's all. So they say what you need to do for these viruses, you need to rapidly diagnose the infection. You have to have high biosecurity, and we need to develop vaccines that are specific for each virus. Passive immunity may be a good approach. They know that works. There are 2 vaccines against TGEV. This is interesting. So there are infectious attenuated viruses. One of them is given to healthy pregnant swine as an aid for preventing. It's a mixed rotavirus TGEV vaccine, and that protects the piglets. And so it has an attenuated infectious TGEV and 2 rotavirus strains. And another one is also given to healthy pregnant swine to provide passive protection to the nursed pigs. Infectious attenuated and subunit PED vaccines have been developed in many countries, but they say they're not good. They don't work well enough to control outbreaks. And for Delta Corona and SADS, there's no available prophylaxis at all. Anyway, I thought that was an interesting overview, and particularly the loss of virulence and the change of tropism of TGEV, and then the neurovirulence of the hemagglutinating virus, I thought are all lessons for other viruses.
And the sparrows.
And the sparrows is pretty cool. Yeah. All right. Now we have a preprint, which is in BioArchive, which is
so open access, but usual caveats apply with preprints. It's not been peer reviewed.
Not been peer reviewed, but this is from a group we well know. It is called the giant Mimivirus 1.2 megabase genome is elegantly organized into a 30 nanometer helical protein shield. This comes from a group at Aix-Marseille University, the University of Grenoble, Leibniz Institute, the University of Oxford. First author is Alejandro Vilalta, and the last author is Chantal Abergel.
And we have 3 co-first authors, Alejandro Vilalta, Alan Schmidt, Leandro Estrosi are co-first authors.
So these giant viruses, it all started with Mimivirus, the first giant virus discovered, and now there is a meeting every few years in Germany on giant virus biology, and Rich has been to a couple of them. Nels Elde went to the last one in 2019. The 3 of us were there. We did a TWiV there. But I went to the first one years earlier and interviewed Chantal and her husband, who worked together on these viruses. And so this is great. This is a lovely structural study. And I just want to tell you all about Mimivirus, because I don't know that we talked about its discovery, but it was accidentally discovered in 1992. People in Marseille and Leeds were working on what they thought was Legionellosis, and they had a potential agent which they did a gram stain on. It looked like a gram-positive bacterium, and it was named Bradford coccus, where the amoeba host for this had originated. Because if you know Legionellosis, the bacterium like to grow in amoeba. So they put it in a freezer and forgot about it. And then in 2003, many years later, the people in Marseille said, let's take a look at that. And it turns out it's a virus. It's not a gram-positive bacterium. So they called it Mimivirus for mimicking microbe. And it turned out to be the biggest bloody virus that we knew of even bigger than pox viruses. And what's the one that in the Jim
Chlorella virus.
Chlorella viruses? They were the biggest ones up until then, right?
Yeah,
but
they were about
dwarfs
300. I think Chlorella viruses are up to 300 kb.
Yeah, well, so this thing, it's got the 1.4 megabase genome. It's half a micron across. So it's not surprising that they looked at it and said, "Oh, that's a bacterium," because it's the size of one.
And it stains in a gram stain too, which kind of threw them. Anyway, so this subsequently, many people worked on this. The structure of it's an icosahedrally arranged capsid, and it's unusual because, well, first of all, it's huge. So the structure was solved, but it's a very big particle, and it's got these fibrils around it. It almost looks hairy, and they have to remove those to do the structure of the virus. And the genome is 1.2 million base pairs of double-stranded DNA, and no one really understood how it was packaged in the particle. And that's what we're looking at here. There's inside the capsid, there's a lipid membrane, and inside that is the DNA. They call this the nucleoid, and that has all the proteins needed for this virus to start DNA synthesis, which happens in the cytoplasm. So as Rich Condit would say, these viruses avoid the nuclear bureaucracy, right?
Yeah.
They do. So they have to bring all the proteins in to start DNA transcription, I suppose, first, right, Rich? And then DNA synthesis.
I have some.
Oh, I was just going to say, I didn't
Go
really
ahead,
know.
Brian.
I didn't know about the fact that they had a membrane-limited nucleoid inside the capsid until I was reading this. And that kind of blew my mind. And I can imagine that someone looking at these first samples, trying to figure out what they were looking at, when they had a gram-positive sample that also had a membrane-limited compartment with DNA in it, must have been quite baffled. And I think of nucleoid as a region in a bacterium.
Yeah.
And so I think that, this made it very clear to me how confusing this would have been to some of the people who first discovered this.
Yeah. So it's actually got two layers of membranes going on here, right? It's an enveloped nucleoid, and the nucleoid has a membrane.
I'm a little, frankly, a little confused about the overall organization of this in terms of where those membranes lie in the whole structure. And, I probably should have spent more time looking this up. Oh, actually, I know.
Well, the capsid itself is not envelope. It's naked, but it's got these fibrils. And then inside are the membranes.
A problem that we run into now and then is that the word membrane can be misused.
Yes.
You guys go ahead. I know where to look this up.
Okay. So these are taken up by amoeba by phagocytosis because they're so big. And then there's a feature of at one five-fold axis of the particle, which was revealed in the structure. It's called the Stargate because it looks like a star. And it appears to open up. And there are structures of opened up particles. And so it was originally thought that that's how the DNA gets out. So the Stargate opens up. And then you have this membrane underneath. Yes, so they say there's a viral membrane underneath the icosahedral that fuses with the vacuole membrane. And then the nucleoid gets in the cytoplasm. Okay, so the nucleoid then, and I guess, Alan, this was your source of multiple membranes. The nucleoid itself has a membrane, right?
So if I just, as a quick look here, look on Viral Zone.
Right.
And look at their cartoon. They've got the capsid. And underneath the capsid, they draw what look like two, they call inner membranes. Okay. And then they have a sort of a fanciful, another thing that looks like capsid underneath that, but I don't know that that's for real. And then a sort of their own fanciful bit about what the DNA looks like. But I think it's suffice it to say that there's, in terms of the DNA getting out, there's lipid in the way. Okay.
Yeah.
Yes.
You not only have to open up the capsid, but you got to deal with the lipid. And they talk about in the paper, a fundamental part of this is one or both of those membranes fusing with an intracellular membrane compartment to dump the nucleoid into that, and that's what gets things going.
So they want to, in this paper, they look at the structure of the DNA. How is it packaged? Because not much is known about that. And so they develop a way, they make a ton of virus particles. They use 12 milliliters of virus 1 times 10 to the 10th particles per milliliter. And then they use that they develop a protocol to open up the Stargate without cells. It just involves some low ionic strength buffer, I think. And the DNA comes out.
And a ritual performed at midnight.
Actually, I think there's 2 steps to this. One is, as you say, a low pH step to open up the Stargate, and that releases the nucleoid.
Right.
Okay. And then there's a second step that involves a gentle treatment with both proteases and detergents.
Right.
To release this, what are they calling it?
Genomic fiber.
Genomic fiber from the nucleoid. Okay?
Right. So they do this protocol on a lot of virus. The particle opens, it releases what they say are 30 nanometer wide rod-shaped fibers, several microns in length, which they call the genomic fiber. And they say they have lovely pictures of this. Complete expulsion of these fibers resembles a ball of yarn, they say. And they optimize this with a specific isolate of Mimivirus. And they have some lovely pictures of this.
I have to say,
right?
I went to figure 1 and I looked at panel B and I said, what is going on here?
Yeah, you could see the virus is opened up,
yes.
right?
It's like Pac-Man sticking its tongue out. It's just, wow.
And then you can see the nucleoid still has its membrane, right?
Yeah.
Right.
And inside is the genomic fiber, which you can sort of kind of see helical character to
Yes.
it, right? Even in that photo. And so they get rid of the membrane, as Rich said. And then you have a ball of yarn, which is in figure C. And this is a long DNA. And then you get higher magnifications. It's quite clearly helical. And they go on to characterize this and do the structure. So they say, okay, there's DNA in here. We can run it on a gel and it's DNA. And there is a fiber, but the nucleic acid is inside this fiber. So there's something on the outside, and it turns out to be protein. So there's a protein shell, and inside is the double-stranded DNA all wrapped up.
Right. So when this thing is helical, we're not talking about the DNA double helix. We're talking about a helical protein structure, and then that has DNA packed inside it.
Right. So the classic filamentous virus structure.
Yes.
Right. Except this is really big. This is a big one, long DNA. So they do cryo-EM to solve the structure of these genomic fibers. And they see a number of different conformations. Then one of them has 5 DNA helices. There's a protein shell on the outside, 8 nanometer thick. And inside there are 5 helices of DNA, 5 double-stranded DNA strands lining the interior of this protein shell. And then there's a channel in the middle, which is about 9 nanometers wide. And then there's another version where they can't really see the DNA because they think it's all been released from the protein. And then they have another version with 6 double-stranded DNA strands and a bigger wide channel, 12 nanometer on the inside. And they have lovely pictures of this. So it's clearly helically arranged, and it's got a protein shell. This is a nucleocapsid. So, in my virology course, I always teach what a nucleocapsid is. A nucleocapsid is a protein nucleic acid assembly that's a sub part of a virus particle. So you think about say a measles virus. There's a RNA protein nucleocapsid inside the envelope. And my students every year say, is there an icosahedral with a nucleocapsid? I said, I don't know of any, because usually it's naked DNA in there, right? And it's not really a nucleocapsid. This, I would say, is a nucleocapsid. What do you think, Rich?
That's exactly what I was going to say. I've been holding back. Okay. They don't call it a nucleocapsid, do they? They call it a fiber
No.
or whatever, but that's exactly my conclusion. Okay, it's a helical nucleocapsid.
Yep.
Or that's what I would call it. It may, it may not be a nucleocapsid structurally and precisely the way we think of a helical nucleocapsid. It's obviously different than, for example, the nucleocapsid for a negative strand virus, a helical nucleocapsid from a negative strand RNA virus.
Yeah.
But given the general definition of nucleic acid wrapped in protein that's inside a capsid, okay, I would call it a nucleocapsid. All right.
Yeah.
Beyond that, in terms of its structure, it's reasonably unique. Now, oh man, I'm getting old, and I've been a long way out because I'm riffing on my poxvirus experience the whole time with this as well, because we use Nassim and I use the word nucleocapsid to talk about the internal structure of poxviruses. And we knew there was DNA, and we knew there was protein there, and we knew there was some structure. The problem is that to get at the structure, the wall of that inner core particle is so tough, and we can come back to this because there's some subtleties here, is so tough that in order to crack it open, you wind up destroying the contents inside any structure. Okay, this is you can spill out the nucleocapsid. You can disrupt the inner membrane and capsid and spill out the nucleocapsid by a fairly gentle, relatively gentle treatment.
So I did the structure lecture a couple of weeks ago and talked about nucleocapsids, and one of the students emailed me, said, could theoretically an icosahedral virus have a nucleocapsid? And I said, yeah, but I don't know of any. And then a week later, this paper comes out. I was so excited to show it to them. What a lecture. So then they say, well, what proteins are in this fiber, right? Because that's interesting. These are presumably viral proteins.
Right. And at this point, this is this complex structure that undergoes these dynamic transitions, and it's in a virus that's got unlimited genome space and thousands of genes. And you're thinking, okay, how many proteins is this going to be made of?
That's right.
And the answer
by the way,
is
mostly one.
Yeah.
But before I do that, sorry, I meant to say this is open access, so you can download it and look at the figures. Figure 2, they have these 3 structures, the 5 double strands, the 6, and the one where they can't see the DNA. And it's just beautiful. You could see how lovely the protein subunits are making up this structure on the outside, and then the DNA strands are inside. It's just, they have a side view and then an end view where you can see the hole in the middle. It's just gorgeous, this thing.
A nice thing about structural papers is even if you don't understand the details of how they got the data, you can look at the picture and get a pretty good idea of what's going on.
Yeah, I looked at this and it was so obvious to me. I'm like, oh, it's like there's a cylinder protecting the DNA.
Yes.
Got it.
Yeah. So they say, what is in this this structure? And they do mass spec. And the main components are 2 enzymes, glucose methanol choline oxidoreductase, GMC oxidoreductases. And these, they say, this was totally unexpected because these proteins compose the fibrils, the hair on the outside of the capsid. And they say later in the discussion, what is the evolutionary explanation for a virus that encodes over 1,000 proteins to use the same one on the outside and on the inside? Which is a good question.
Yes.
Right.
Well, if it ain't broke.
Yeah, it worked in both cases.
Worked.
And so what they're doing, they later speculate that maybe as this virus is moving through the phagosome of the amoeba, this is a pretty extreme environment with a lot of oxidation going on. Maybe these are protective somewhat. So they don't know, of course, but that's their speculation.
My read on this is that they have no evidence that this is actually active as an enzyme. Okay.
Right.
And which I think is just as interesting as it being active. as if the structure was co-opted for this purpose. I know of at least one other example of that kind of thing happening, which is vaccinia virus, where one of the membrane proteins has just whopping homology to carbonic anhydrase, right? Which is a protein that interconverts CO2 and carbonate, controlling pH balance in the blood. Okay, and as far as we know, it has no activity, and this protein is homologous to carbonic anhydrase, but it has on the carboxy terminus a membrane anchor. Okay, so it's taken carbonic anhydrase, added a membrane anchor, stuck it in the membrane, and it's involved in attachment. So that's a good example of what apparently somewhere in evolution was an enzyme that's been co-opted for a structural role.
I like
and has nothing to do with...
It's kind of like deciding, okay, we need to put a body on the outside of the car. Let's build it out of wrenches. We've got them. So let's,
But here in this case, if you look at the pictures, these lovely tubes, the protein that make up the tube repeated over and over again is this oxidoreductase. So maybe it just structurally worked, right? And it doesn't matter that it's inactive. I guess what they could do is, look to see if the protein's active and knock out the active site and see if it still forms these structures, right?
Right.
Will be cool.
I guess you could first actually just purify it and see if you could get enzymatic activity in a test tube.
Well, they kind of have it pure with these genomic fibers, I guess, right? So they could do that. But this is a viral or cellular protein?
Which? I think it's a viral protein.
It's a viral protein.
Yeah.
Okay. But there are cellular.
There are cellular proteins in this family. Yeah. And their job. I spent some time with this because, when it comes to oxidation reduction, I break out in a sweat. That was never my strong suit. Okay. But so I had to dive into this a little bit. They say that this is the family is GMC oxoreductase, which is glucose methanol choline oxidoreductases, which you can look up in Wikipedia, my favorite scientific journal. And what they do primarily is reduce primary alcohols to aldehydes. Okay. And they use a FAD as a cofactor because there's electron transfer involved, which is flavine adenine dinucleotide. Oxidoreductases in general do lots of different things, including energy transfer. But this is not one of those. This is doing basically a chemical reaction to reduce an alcohol to an aldehyde. But they don't know what this particular one, and there's a bunch of them that do these reactions to a bunch of different substrates, okay? In this particular case, they don't even know if this thing is active, and if it is, they don't know what it would do.
Right.
I bet it's not active.
I bet it's not active. Yeah. I bet it was just grabbed because it fit.
Yeah.
Yeah. Okay, so then they do this interesting exercise where they say, okay, we know how long this DNA is. What has to be done to fit it into this nucleoid? The nucleoid is, so the genomic fiber is 40 microns in length, and they know, there are 5 or 6 strands of DNA in the fiber. In each fiber, there's 5 or 6 strands put in there. They must be folded back and forth on themselves. And so they say it can't just be linearly added because it would be too long. And so they say the genome is probably organized as a shell. This nucleoid or this genomic fiber is this helical shell. It's internally lined with folded DNA strands. So you have to think that the DNA is folded and they're helically or regularly arranged inside, but they're folded back and forth. That's why you have 5 or 6 strands, because the genome is going back and forth in this 40 micron long nucleoid. It can't just be one strand of DNA. It wouldn't fit. So 5 or 6 would accommodate it. So that's brilliant, right?
Yeah.
So a nucleoid of so length, you got the DNA going back and forth inside this tube. It's gorgeous.
It's elegant.
And they say, which I didn't realize, there's an archaeal virus that has a similar structure.
Yes.
I didn't know that. So, it's repeated. If something works, you reuse
It'll
it.
happen again.
And then I think the final part, which I think is also very cool, they say, okay, what other proteins are here besides these oxidoreductases? Well, they find a bunch of proteins involved in RNA synthesis, mRNA synthesis, because remember, this genome gets in the cytoplasm, it starts to make RNA proteins, and then starts to reproduce its genomes. There is a bunch of subunits of RNA polymerase. And they say, we look back at the EMs. Upon close inspection of the negative stain, they say macromolecules strikingly resembling the characteristic structure of the poxvirus RNA polymerase were seen in the EMs of their genomic fibers. And so they end up modeling the RNA polymerase in between this cage of multiple DNA strands. It fits right in there. It's figure 4, right? So you have the outer protein shell made of these oxidoreductases. You have 5 or 6 DNA strands, right, going inside. And then every so often an RNA polymerase sitting in there. Don't you love that, Rich?
Yeah. And I got a long rap about this too, but go ahead.
No, that's all. Rap. Go ahead. Tell us what you're thinking.
So, there are several viruses that package transcription enzymes. I don't know quite where to start or how to go about this. Okay, but let me just do the vaccinia problem.
Yeah.
Okay.
Yeah.
So vaccinia, a pox virus, it's different than this, but has some similarities. Okay, where the important thing is that the entire life cycle is cytoplasmic. So it has to package an RNA polymerase that it delivers along with the genome and then do early transcription right away. Okay.
Right.
In the case of pox viruses, underneath the membrane, the pox viruses have an outer lipid membrane, a proteolipid membrane. It's got a lot of protein in it. And inside of that is a core. And they uncoat and dump the core into the cytoplasm. And the transcription goes on apparently inside the core. The core doesn't dissolve. Okay, goes on inside the core, and the RNA is extruded from the core in an energy-dependent fashion. Then you make some early viral proteins, one of which turns back and is, at least one of which, is important in then disassembling the core, at which time the DNA can decompress, if you like, compact, and becomes available for DNA replication. So you have to have the RNA polymerase and the DNA organized inside. It doesn't have to be organized, but I assume it is.
Yes.
Inside this core in a fashion where without a whole lot, there's a little swelling of the core, but without a whole lot, you have to be able to do all the movement that's involved in doing transcription and extrude this stuff out through the core. And it's been it has been and still is a very troublesome problem of how that happens. The other thing that comes to mind when I think about this, and they mention this in this paper, is reoviruses. Double-stranded RNA that's contained inside a protein capsid,
Right.
and is really never completely unencapsulated.
Right.
Contains an RNA polymerase that copies the RNA genome. And in fact, a lot of the enzymes are part of the inner shell of that particle.
Right.
And it does the same thing where it does transcription and then extrudes the RNA through pores in the capsid. And there's even these just brilliant electron micrographs from decades ago of these cores that if they're active in transcription, you get all these hairy projections, which is the RNA coming out of the capsid. We were always, we mused about trying to do the same experiment with vaccinia, which should be done sometime and has never been done. So how you organize the nucleic acid inside a shell like that in a fashion where you can do the transcription without uncoding has always been a mystery and a fascinating mystery. This is a little different because this guy gets dumped out, apparently, from what I can understand, and decompresses some. It's got the DNA. It's clearly from this article in a nucleocapsid. It's got the RNA polymerase associated with it. Okay, but at least some of the constraints of a larger encasing structure are removed so that this thing can relax some. That's a little easier for me to imagine how then you could transcribe in an early factory. They also speculate, as has been speculated with some of these other viruses, that the RNA polymerases are actually perched on early promoters, ready to go. And in the case of the pox viruses, one of the interesting observations is that if you put a mutation in an early transcription factor that is responsible for binding RNA polymerase to a promoter in the DNA, and you infect a cell with that thing under conditions where, I mean, you can make this virus and infect cells with it, it won't package DNA. If it can't package the RNA polymerase, it can't package the DNA. So the implication there is that the RNA polymerase is actually an important part, at least structurally, if not actually actively, in creating that, in doing the packaging and creating that nucleocapsid core. And my guess is the same sort of principles are involved here.
So I think a lot of that is really fascinating, Rich, and there's a lot of information there I didn't know and is really cool. I often spend a lot of time talking to my students about how nucleic acid has to be organized in some way, and I make them think about what happens if they throw their headphones in their backpack. Which is quickly becoming a really dated example, as now they all have AirPods that just don't have wires, and how you have that tangled up headphones that you have to spend a long time taking apart. But what I was thinking about as you were just giving those examples, Rich, is that there is sort of a very similar example that's being talked about a lot in the HIV field, which is that HIV is thought to enter and have its conical core stay relatively intact and have reverse transcription occur in that conical core and then deliver the reverse transcription product to the nucleus. And the idea is that that's protecting the nucleic acid from being sensed in the cytoplasm. One thing that I've always wondered in looking at that is, how does the capsid know it's been uncoded? What is the conformational change that says, okay, you can start doing reverse transcription now? And it sounds in rotavirus or reovirus, I think you had mentioned, and reovirus, and in vaccinia, it's a very similar problem, a very similar process that's happening. And so maybe this is actually a broader phenomenon than we thought.
I completely agree. Yeah. Okay. So first of all, that you're not just stuffing headphones in your backpack in any situation. That my guess is that in all cases there's an organization, there's a strategy to packaging DNA, and it becomes, or RNA, packaging nucleic acid, and it becomes, in my mind at least, particularly problematic when the requirement is to package it in a form that has transcription potential. Okay, that's sort of another layer of complexity on the whole thing. In particular, if that transcription has to go on inside a particle that is not, as far as I can tell, changing a whole lot in terms of its shape. I see packaging DNA as being, really ramming this stuff in there. Okay, there's not a lot of room to move around, and yet there has to be some slack in the system because as RNA polymerase transcribes DNA, there's a lot of movement.
Well, and it may
at least
be
relative movement.
It may be that the signal for the you've been uncoded, as Brianne was pointing out, because that's a problem that the virus
Right.
needs to sense somehow. And it may be that the slight relaxation of the core that allows enough space to bootstrap the transcription process may be the signal.
Well, clearly in the case of Vaccinia, well, actually in the case of Vaccinia and reoviruses, in the case of, there's I think in a broad sense, both of those viruses can be conceived of as having two structural layers, okay? And it's removal of the outer layer that seems to activate the core in the case of both reoviruses and Vaccinia, okay? When you remove the when you deliver the particle into the cytoplasm and remove the outer membrane and release the core, that activates it, okay? Now, there's probably a whole lot more to it than that. But, and in the case of reovirus, that's a double-shelled virus. And it's when you get rid of the outer shell that you now activate the inner one. We've got to get Sarah McDonald back on the show. To go over this whole thing with us.
Well, so notably, we're talking about a virus here that has that inner layer of that membrane.
Yes,
right.
And I think, coming back to the paper, one of the important things here is that this is insight into one way to package DNA in a particle like that, along with RNA polymerase. Okay,
that's right,
and more detailed insight than I think we have with any other DNA-containing virus.
Yeah, I agree. It's a principle now. It really is. And so they mention here, we should have mentioned this at the top. So you have the capsid, then underneath the capsid is a membrane, and then around the nucleoid, there's the other membrane. So the first membrane is just sitting below the interior of the capsid shell. Okay, that's right. There are two membranes, and that's how they're arranged. So I think the last thing I want to say is this. This sentence. Last sentence of the paper. The parsimonious use of moonlighting GMC oxidoreductase is playing a central role in two unrelated substructures, the fibers and the shell, the shield for the DNA, questions the evolutionary incentive leading to such an organization for a virus encoding close to 1,000 proteins.
Why did you simplify here? Yeah.
Yeah, it's just this gene saying, I got this.
Yeah.
Okay. I don't care how many proteins you got. I got this. I got these two jobs. I'm doing them. You get out of the way.
Right. That's very cool. I think this is a really good paper.
Actually, it'd be interesting to have, I don't know if it exists, this kind of structural information on the fibers.
Yeah.
So I wonder if the structural dynamics of the fibers are similar to what's in the nucleocapsid.
It would be interesting to take these genomic fibrils, Rich, and see if you could do in vitro transcription, right?
Yes, it would. And I'm sadly lacking in my Mimivirus literature here because, in the case of Vaccinia, you can uncoat the virus just with a little neutral detergent and drop the cores in a test tube with nucleoside triphosphates, and it makes early RNA and caps it and polyadenylates it. Okay. Cool. And I think the same happens with reoviruses as well. I don't know what the situation is with Mimiviruses, whether those sorts of experiments have been done. I would imagine that you could, but I don't know.
Mm. So after Mimivirus was discovered, the same team found a slightly larger virus. They called it Mama virus. And then they found another one. They called it Mumu virus. They have a great sense of humor in naming. At the last giant virus meeting, Matthias Fischer sang a what he called a viral opera. And one of the verses was sung to the tune of Bohemian Rhapsody. And the lines, remember this, Rich?
Yeah.
This was just great. Mama just killed a cell. All I wanted was a feast, and phagocytosed a beast. Mimi, life has 4 domains, but what remains is just a truck. Moo moo, ooh, ooh, ooh. He's saying it perfectly. Neither fish nor fowl am I. If I'm not classified this time in 2 years, carry on, because ICTV matters. You have to sing that to the Bohemian Rhapsody. It was just perfect. I loved it. All right, let's do some email. I have to run off and do something for 5 minutes. So, guys, do some emails, okay? Alan, you can take that first one.
Okay, I will take the first email. This is from Christine, who writes, Dear TWiVcasters, in response to John Udell's comments about the need for science education at a young age, TWiV 857, I'd like to give a shout out to the School for Science and Math at Vanderbilt, a program for public school high schoolers here in Nashville, a classroom full cohort each year who spend 1 day a week at Vanderbilt University learning science. My daughter Alice was lucky to be admitted to this program. She's 23 now, a landscape architecture student at the University of Tennessee at Knoxville, and it is on her that I rely to troubleshoot ideas about all kinds of topics since her critical thinking skills are so well developed thanks to her 4 years as a baby scientist. In 8th grade, she was hesitant to apply to the SSMV program since it meant more work in high school, but she came to appreciate it very much and made some lifelong friends there. Still, while Alice and her friends are lucky to have been a part of an advanced, high-quality science program in high school, it would be better if more students, or all students, were exposed to the ways of thinking and methods of science. My own exposure occurred much later in life when I was hired by a science publisher, VCH Publishers, later part of Wiley, and worked there for a few years in close contact with chemists, biochemists, physicists, molecular biologists, and Buckminster Fullerinologists. Buckminster Fullerenes, edited by Drs. Billups and Ciuffolini, was published by VCH while I worked there and provides a link. It was striking to a literary person such as myself how large the world of science is, and yet literarily inclined people such as I had managed to ignore it most of the time. I do my best now to publicize TWiV, especially episodes that are easier to follow for lay people via my Twitter feed, Irisaurus, and among friends. I'm not always successful in convincing non-scientists to listen to TWiV. It takes some insisting, just as it took some work to convince Alice to apply to SSMV when she was a teenager. Well, I'd like to say that this pandemic is an opportunity to strengthen science education by any means available. Thanks for all you do. I usually cook or bake while listening to TWiV. Your back-and-forth conversation and comments have inspired some delicious soups and baked goods, if I say so myself. Warmly, Christine, and I hope you're making some nice ribs down there in Tennessee with your porcine viruses episode. Thank you, Christine.
Hopefully, some bacon.
Yes, some bacon would go well.
What I get out of that, among other things, is that as she talks about her daughter's experience in that program, she says nothing about the specific science. She talks about critical thinking.
Mm-hmm.
Yes.
That's the point. And it's so frustrating because I think kids can be turned off to science because they're not taught to think critically. They're just taught to memorize a bunch of facts.
Yes.
Okay. With no reason for it or anything like that. And that's not the point. The point is to learn how to solve problems.
Yes.
Learn how to think about stuff.
And I like that she mentions that her daughter is now a landscape architecture student. And so I like the idea that we can point out that these skills are not just useful to budding kind of bench scientists, but those critical thinking skills can be useful for a lot of people in a lot of ways.
Yes.
And Christine, since you're cooking while you're listening to this, I think we've talked about this before, but I think you should just Google spiced bread egg in a hole.
Oh, yes.
All right. And you'll get a recipe from the Washington Post that has become one of my favorite breakfasts.
All right.
You call the shots here, Alan.
Okay. It devolves to me, right? So we're going by seniority. All right. So that was Christine from Nashville. And how about Brianne? You take Anthony's.
All right. Anthony writes. Anthony gives us a link to an article in Nature and says 3 key theories. For now, whichever idea a researcher favors, quote, often comes down to gut feeling rather than any sort of principled argument, end quote, says Richard Nair, a computational biologist at the University of Basel in Switzerland. They are all fair game, says Janal Beeman, a medical scientist at the National Institutes for Communicable Diseases in Johannesburg, South Africa. Everyone has their favorite hypothesis. And I am guessing that the 3 key theories here are where Omicron has come from. Yes, that's what the article is about.
Yeah, that's the title of the article. Where did Omicron come from?
Yes.
3 key theories.
And so thinking about things like the spill back from a different animal or being replicating in an immunocompromised person or this sort of cryptic spread. And so these quotes say that, there's some gut feeling and it's hard to decide between the two of them. Anthony writes, distressing twice. 1, that the origin of SARS-CoV-2 is shrouded metaphorically in the mist of communication with China and literally in the midst of deep jungles is understandable. What defies comprehension is that the most studied thing on the planet has left everybody clueless. 2, the confusion with the concepts of theory, speculation, observation, inspiration, interpretation, and experimentation, hypothesis. There are very few theories: evolution, gravity, relativity, quantum mechanics, etc. Theories are the closest thing we can closest that we can experience truth. Any gut feelings on theories mean nothing. I think that realistically here, what we really need is a lot more evidence. I don't know that this is going to always be leaving everyone on the planet clueless as the most studied thing. But I think that right now, only a few months after Omicron was first described, we don't yet have the evidence to have some kind of definitive answer. And as yet, I think a lot of us are using our best guess or our most educated information to try to come up with a hypothesis. And if I were to tell you that I was thinking about something here in terms of my gut feelings, I would be trying to highlight the fact that I don't actually have specific evidence that's making me lean towards that hypothesis, that I'm using sort of an educated use of knowledge, but that I don't really have a ton of additional evidence behind it. And so for me, that would be actually a way that I was couching uncertainty, not certainty.
Yeah.
There's just no way this is going to be figured out, as you say, in a couple of months. No. Right? To think it's left everybody clueless ignores the fact that it takes a long time to figure things out in science. So I don't think that's fair.
I think we're not going to get a good fix on where Omicron came from until we're a few more variants down the line. Maybe get past pi and whatever else is coming next. And I think we may stop naming variants of concern as they become of less concern. But we need to see this process happen again, because I think we missed the opportunity to see it happen in this case.
Yeah. How long did it take to realize that SARS-CoV-1 came from bats and civets?
Yeah.
That was not months.
Right.
But it's also, for this fellow to say it's a gut feeling, I think is incorrect.
Right.
It's not just a gut feeling, because there is that AIDS patient in South Africa who had 9 months of SARS-CoV-2 infection and who spawned some of the similar mutations. That's not a gut feeling, right? There's the phylogenetic analysis that we talked about, and also the sewage, the wastewater analysis that suggests there are very different changes in rodents. It's not a gut feeling. I think that's completely wrong. And this is where you get in trouble when you talk to someone and it's a gut feeling. And then all of a sudden everyone's saying it's a gut feeling. And then Anthony gets offended. So it's not a correct way to look at
Yeah,
it.
what we call gut feelings are usually informed by a bunch of
Yes.
science.
Yes.
Okay.
Yes.
So we have to be careful with our vocabulary.
There are
Exactly.
data here, just not enough to say something definitive,
Yeah.
and so we can say, okay, the data we have suggest these theories, and we can't narrow it down much more than that.
And I feel as though the data is stronger for this theory than another.
Right.
But I know that I am not certain about that.
Yes.
And I get to change my mind.
Yes.
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 Oh yeah. Oh yeah. Oh yeah. So I guess Rich is next,
Yes.
right?
John writes, Doctors TWiV, mostly sheltering in place here in Greater Braddock, awaiting the advent of 5C tomorrow and the double digits predicted to herald the arrival of March. At the end of 871, talking about Maurice Hilleman, you mentioned him as having saved the lives of more people than anyone ever. Not to question that, but since Norman Borlaug was also mentioned, it might be an idea to divide the honors along the lines of the Four Horsemen. So Hilleman could have have saved more lives versus plague, broadly considered. For Borlaug, it would be versus famine. And I would propose splitting that category into indirect and direct categories. Borlaug would win in the indirect line for the Green Revolution. But for directly feeding people, there's someone that nobody thinks about anymore, Herbert Hoover, who mobilized and orchestrated food relief efforts during and in the aftermath of World War I and also after World War II, a task he largely rose to without being asked. Hoover's efforts during World War I have been covered in various places, probably best in his 1951 autobiography. But for the World War II efforts at the behest of Harry Truman, the only really good account I know of is Herbert Hoover and Germany, Louis Lochner, 1960, that I recently read, and which also summarizes the World War I efforts. It's a relatively short read, too, and if it encourages someone to read his autobiography, all the better. Lochner basically just lays out all of the details, while the autobiography is written with a certain degree of dry wit and contains the occasional bit of comic relief. Lochner details how the World War II relief efforts came about after the Russians reneged on their agreement in the partition of Germany. The products of the different sectors were supposed to be shared equally amongst the sectors. East Germany was the breadbasket, but instead of distributing the harvest, the Russians took them for themselves. So a massive food relief effort was mounted actively and vigorously, headed by Hoover, who was then in his 70s. If you like, that's my pick. And that makes a good segue to the situation in Ukraine that I'm following closely. When I saw that Jon Stewart's podcast on that today, I immediately thought you might like it. 3 comedians cheering on one of their own, Zelensky. Did you know that in his earlier career he's a comedian? It's an excellent and only 16 minute 16 minutes long. And Stewart, by the way, started off as a chemistry major at William and Mary, class of 84. I didn't know that. He gives a link to the YouTube video. And he's got a footnote to Norman Borlaug, who was a corn geneticist, Charles Burnham's graduate student. Burnham co-founded the American Chestnut Foundation that I've mentioned before with encouragement from Borlaug, and was himself a fellow graduate student with Barbara McClintock and George Beadle, seen in this wonderful 1929 photo of future luminaries in genetics. Roland Emerson's early work had recapitulated Mendel's, which hadn't been rediscovered at the time. So he was by this time already somewhat luminary in that category. And he gives a link to a picture here that indeed has, wow, Cold Spring Harbor Laboratory picture with Barbara McClintock as a graduate student with Charles Burnham, Marcus Rhodes, Rollins Emerson.
George Beadle.
kneeling with his dog. Holy cow. And what a crew. Look at them in their knickers.
That's very cool.
That's cool.
It's very cool.
It's wonderful. So I did not know this about Herbert Hoover.
No, I didn't either.
Very interesting.
Yeah.
Cool.
Thank you. Appreciate that. All right. Then we'll wrap this with Philip. As a watcher of TWiV, I understand by conventional wisdom an HIV vaccine would need to be 100% sterilizing, which is a high bar to reach. So us older folks had chickenpox that leaves the virus in us, and it later activated to give us shingles. I would summarize that HIV must be more deeply hidden in the body than the zoster herpes virus. Is my thinking correct? I had the earlier attempt at a shingles vaccine, which I understand was the chickenpox vaccine. I did the first dose of Shingrix and forgot the second, so I started anew and properly completed the 2-dose series. More deeply hidden. I don't think that's a good way to look at it.
It almost seems like he's thinking that HIV doesn't get later activated and herpes does. But that's not correct.
It's not an issue of depth of hiding. It's an issue of consequences on return, I think.
I would say not only consequences of the return, but frequency of the return.
So HIV,
because
it persists in an ongoing infection, whereas zoster retreats and goes dormant completely.
Yes,
and is not doing anything for decades.
Okay, so here's what he's thinking. He says, okay, the Shingrix vaccine is going to keep me from getting shingles, but it's not getting rid of the virus.
Right.
That's right.
Okay, the virus is reactivating, but the vaccine-induced immunity is keeping it down so that you don't get the rash of shingles.
Right.
Okay, so it's not getting rid of the virus, and maybe that's why he said HIV is more deeply hidden. So that's not it.
Right.
No,
but an HIV vaccine that could accomplish the same thing as Shingrix would be transformative, right? If it could keep if it could force the virus not to do its thing, force it into some kind of a dormant or boxed in state, then that would be effective. It's just it's not really the same biology because HIV is not in a dormant state.
From that point of view, we have often said that an HIV vaccine has to be sterilizing, but maybe that's an overstatement. Okay,
right?
Because if you could create an HIV vaccine that worked the way you described, Alan, such that it could keep the reactivation under control, that would be fine.
Right.
Yeah, then you're just arguing about the definition of the word vaccine.
Yes.
A problem with HIV is that it's throwing off immune escape variants at an alarming rate. Okay, so such a vaccine would be very difficult to do, whereas a varicella zoster is basically stable, right? I don't know of antigenic variation. In that.
I mean, Brianne, I would say it's a vaccine because it prevents disease, right?
Right. Well, it would be a vaccine against AIDS later on, or against AIDS reactivation, not a vaccine against the first acquisition.
Not a vaccine against HIV infection. Right.
Yeah.
But that's the same for all vaccines. They don't prevent that acquisition, so it's not really. It's just that the peculiars of HIV are a problem. But if you can prevent disease, that would be fine. Yeah. Right. Well, if you could prevent immunosuppression, that would be fine.
Yeah, I mean, because the shingles vaccine is not preventing you from getting infected. It's just preventing you that reactivation.
No. Right. That horse is already out of the barn.
Disease.
But
exactly.
It's preventing disease, right? And there is some virus produced, but there is no disease.
And I really want to know, Philip mentions the older folks had chickenpox. Does that qualify me as an older folks? I don't know how I feel about that.
older than the young folks who got vaccinated for chickenpox.
Exactly.
I guess so.
And my sense is that, varicella virus, the latent genome, is probably kicking out, reactivating at a very low level frequently. Okay.
Yeah.
It's just a matter of keeping that tamped down. And that's what the vaccine
So
does.
in fact, you could take anyone with AIDS and give them this vaccine, and it would prevent them from developing immunosuppression. Maybe they would not have to take antivirals.
Right. The vaccine would
Potentially.
basically substitute for the drug cocktail, would be doing the same type of thing, is
Right.
the theory.
Yes. Yeah, in theory.
All right, let's get
but
on
we-
it.
Yeah, exactly.
Just a couple of other things, just to clarify. He says the shingles vaccine, which I understand was the chickenpox vaccine, yes, given at a higher dose. That was the original vaccine.
The original one, yeah.
The new one, Shingrix, is a subunit vaccine. It's a glycoprotein from the virus.
Yes, exactly right. Okay, let's do some picks of the week. Brianne, what do you have today?
So I'm scrolling. So I have an article that I found really interesting called "The Dinosaur Age May Have Ended in Springtime." So this is in the New York Times, and this actually looks at some work that people have been doing where they're able to partially figure out about when in the year the extinction event happened that killed the dinosaurs, based on looking at when different organisms were coming out of hibernation in the northern hemisphere, when young were being born. And so one of the people who's quoted here hypothesizes that it is in April, or that was in April, but they have a bunch of data that they talk about that is leading towards thinking that about this as being spring in the northern hemisphere. And just the ability to narrow things down in that kind of way, and the way that they talk about how the different types of data have been used to support that, I thought was just super fascinating.
That's cool. Amazing, right?
This is a great article.
Yeah.
That talks about discovering, kind of pinpointing when the meteor hit.
Yeah, and using some samples in North Dakota that were found.
Yeah, of a sudden catastrophic die-out.
Right.
And figuring when that was. But scientists have known when the meteor hit just over 66 million years ago, give or take 11,000 years.
Yes.
Right.
Yeah, we're still not pinpointing it to year, but this gives you something that's more evocative in a way.
Right.
Right.
You know, it was,
What season it was.
It was in the springtime.
Rich, what do you have for us?
I'm just throwing this out there. It's been sitting in my box for a long time. The movie 12 Monkeys.
Yeah.
Okay. And I need to watch this again.
This is a
For
great
anybody who hasn't
film.
seen this, I think this is one of the best.
Yeah.
One of the best things going, because it's relevant to TWiV, because it's about somebody trying to unleash essentially a pandemic, and it's got all sorts of time stuff.
Time travel.
Right?
Yes.
What is it? It's about people in the future where this pandemic has wiped everybody out, trying to send somebody back into the past to prevent
Yes.
it. Okay? And it's got all these crazy, twisty time things and a bunch of crazy, twisty plot things.
Yeah, the mechanism of time travel is such that they have to use somebody who had an ancestor who was in the time that they need. And so they pull this convict out of the underground prison in the post-pandemic world and then send him back. And it's set in Philadelphia.
That person is
Bruce Willis.
Bruce Willis.
Yeah.
And it features one of the main characters is Brad
Pitt.
in just an incredible role.
Yes.
As this crazy dude. It's wonderful.
Yes.
I've never seen it or heard of it.
Okay.
Yeah.
Well, I'm glad I picked it then, because you got to find the time to do this. It's a crazy movie.
When was this made?
1995.
Wow.
I've heard of it, but haven't seen it. And I'm now wishing I didn't have to go grade some exams tonight.
Yeah, this is definitely worth streaming or renting or whatever.
All right. Alan, what do you have for us?
I have going to toot my own horn a little bit here, but also a book that I recently reread, and it's free. It's on archive.org. Was published in 1921. It's called The Track of the Typhoon, and this ties into our sailing arc here on TWiV. So William Washburn Nutting was the editor of Motorboating magazine in 1920, and they did motorboats and sailboats. And he and a couple of friends who, if you're up with your boating history, you may recognize some names. He called a buddy of his, William Atkin, who was a legendary naval architect, designed a bunch of boats around that time, and wanted him to design a boat for a transatlantic crossing, which was not just, you pay your money to British Airways and you fly. This was a big deal to cross the Atlantic in 1920 in this, what was at the time, a tiny little boat, 45 feet long. So it's an insane adventure. These guys get on this boat and sail across the Atlantic to go watch the yacht races off Cowes, England. And then they bop around Europe a bit, and then they sail back. They almost die several times. They nearly starve to death at one point because they didn't lay in enough supplies in the Azores. It's well written and fun. There are a couple of parts where it drags a bit, where he's just inserting log excerpts, but it's interesting for boat geeks like me. So this has gone into the public domain because it was published in 1921, and it is, if you would prefer to hear someone reading it in a familiar voice instead of reading the book yourself, you can now do that. This was my first complete book as a solo recording for LibriVox. So there's a public domain recording of me reading The Track of the Typhoon.
Cool. Excellent.
Go grab that.
So did you volunteer to do this, or did they?
Yes, this is a pro bono thing. It was. I was looking around at, what are public domain books that have recently come into public domain that I would actually want to read aloud. And I recalled this from an obscure magazine that I get called Messing About in Boats. They published it in serial form a couple of years ago. I said, hey, that would be fun to read. And it was.
How long did it take you to read it?
It was, I split it up over a period of a couple of months. So I'd go and record a chapter, and there's some process, there's audio processing you have to do, and then you upload it. And LibriVox is a whole procedure. There's a proof listener who then listens to make sure everything is up to spec. And so I did it a chapter at a time, and every week or so I'd do a chapter, and then toward the end I kind of pounded them, a few a week.
Did they invite you to read a book, or did you just contact them in the beginning and say, no, right?
So I picked LibriVox some time ago as a nifty little project. It's a group of volunteers who read books that have gone into the public domain and make audiobooks out of them, which are then also in the public domain.
Cool.
It's just a community of open source hippies online who have put this together, and they release their stuff on archive. And you can either join a project somebody else is doing where they're having multiple readers do a book or a play, or you can pick one yourself. And I decided to pick this myself.
So one question: as you're recording, you're gonna goof.
Yes.
Do you stop and back up?
There are two ways to do this. You can either record in a continuous run and make some kind of an easily identified noise. When I first started doing it, I had a pen that I would click a couple of times into the microphone that would leave distinctive spikes on the audio track. And then I would go in and clip out after the spike until the next clump of sound, and that takes some time in the editing process. But you don't have to do anything. You don't have to intervene in the recording. You just, click, click. Okay, take two.
That's a good idea.
You could tap the mic.
Because I often I know that we make a mistake and I want to edit it, and I can't find it. And I have to listen a lot. So that is a very distinctive, wow. Yeah. That's a good
Anything
idea.
Anything that'll put a good spike on there that you've got in your digital audio workstation.
That's a good idea. What a good idea. Okay. Thank you for asking, Rich. I've learned
the other way you can do it is something called punch and roll, which I switched to doing after a while, where you have some kind of a flub, you stop, you back up to you cut at that point, and there's a setting in recording software where it will then replay the last X number of seconds and then start recording from there. So you get in your headphones yourself reading that same text leading into the mistake. You can then reread it and synchronize with the timing, and then you pick up right where you left off. And
that makes more sense to me.
That's less editing, but that would not work so well for TWiV.
Right.
No. No. Cool. That's very cool. All right. So my pick is another of my YouTube channel picks of creatives, right? I've had a number of cooks and an engineer who did cool stuff and others, musicians. This one is Anna Mason's art channel, and she is a watercolorist who, paints for you and shows you how to do things. And she is a realist, so she paints flowers and animals and fruits, and very realistically. And I mean, I don't want to paint or learn to paint, but I just like watching it happen. I really appreciate, like people cooking and doing creative things and having talent. And she obviously is incredibly talented. She starts with the very light pencil drawing of what she wants to do, and then she paints it in, and she's so good. It's amazing. I've looked at a couple. There's a red poppy, which is amazing, and then there's a realistic strawberry, which looks like a bloody photograph in the end when she's done with it. It is really good. So I appreciate her skills, but also the video production is really high quality, right? She is, looks great. She has great lighting. The editing is perfect. She has a camera on her and a camera down, of course, and the cuts are perfect. She doesn't spend, 3 hours painting a little area. She just gives you an idea, and then she moves on to the next one. So it's beautifully edited, so they're not too long. So I really like it.
Very cool.
It's somewhat soothing to just see people create things.
Yeah,
right?
she's like you've watched Bob Ross videos, right?
Sure.
Yeah.
Happy little clouds.
Happy little clouds. Happy little trees.
All right, we have a couple of listener picks. First one is from Richard, reporting from the South Island of New Zealand, where it's currently 17C63F, with the heavens delivering an almighty deluge. As a farm animal veterinarian, I've been fascinated with the detection of SARS-CoV-2 in all manners of animal species, especially in white-tailed deer, as discussed in 847. Please see the link provided below for my pick. If I could be so presumptuous, I keep hearing Vincent and Rich's voices in my head when looking at the map of distribution of detections reported in animals. We need to get out there and do more sampling. I immediately question whether the perceived prevalence of reported outbreaks in North America and Europe are because they are doing the most sampling if compared against other parts of the world, such as India. In the species I attempt to specialize in, cattle, one recent survey even tested 1,000 cattle in Germany, where seroconversion was detected, albeit in only 11 cattle, which were most likely exposed to by their handlers, and the authors couldn't see evidence of cow-to-cow spread, reassuring for this dirty cow vet. Even though this virus has caused so much devastation across the globe, I cannot help but being completely fascinated by it, and your infectious enthusiasm has not been as effective as Paxlovid to help alleviate this malady. Thank you for completely reinvigorating my interest in emerging infectious diseases, so much so that I am now going to begin a Master's of Science in One Health, as I believe in this concept. I believe this concept, One Health, is critical for increasing our surveillance and understanding of what is out there. Yours sincerely, a fellow Rich.
So this is cool. SARS-CoV-2, the link is to an article called SARS-CoV-2 in Animals, a Situation Report, or Rep 9. They have a nice table here that shows where they have found, we were talking about this earlier, virus, cat, dog, mink, otter, pet ferret, lion, tiger, puma, snow leopard, gorilla, white-tailed deer, fishing cat,
Binturong, yeah.
Quadamundi, spotted hyena, a lynx, a hippo, and a hamster. Now, a lot of those species are related. Okay,
not the hippo and the hamster.
That's a lot of crippers. Yeah, the hippo's out there.
So this hippo is only in Europe, right? Must be a zoo.
I suppose.
They can't say exactly where because that would be a hippo violation.
Yeah, I guess this is PCR, right?
Don't know.
That's a good question.
They say number of outbreaks reported by species and region. It may vary.
This is pretty cool.
Yeah.
I didn't know this. A sit rep for non-humans. That's cool. And then a link, then relevant links at the bottom. Very good. I like this
Excellent.
very much.
Cool. Now
Cool.
they just have
Excellent.
to tell us about sparrows.
Yes.
Sparrows, indeed. Richard is a large animal veterinarian.
That's a veterinarian who
Very
works on
cool.
large animals, not a...
Yep. Right. And Jason writes, I'm a chemist by training and computer nerd professionally. Remember my message about home and lab automation, which got read on air in Epitope 821? So I found a nice crossover between the two, which may be helpful for researchers who need to count things as part of their daily work. The Apple App Store has a number of apps which automate counting. And I'm sure the Google Play Store does too. Just aim the phone at the things which need counting, and AI in the app does the rest. All the apps I've found need a license, so I don't have any real experience with them, but they look promising. And he gives 2 links. So what if you had like a bunch of marbles on the table? You could count them. Is that the
Yeah.
idea?
Vincent, plaque assays.
Yes.
A plaque assay. Wow. That would be so cool.
You point
Yes,
your
Amy.
camera
Point your
at the
camera
plaque
at the plaque
assay and it
assay.
counts them.
Amy, you listening? Let's try it.
So what you're telling
She's
me is
always
if
like,
I
I can-
run into the lab right now and get a plate with some colonies on it, I can colony count this way?
That's
Yes.
what they, yeah.
Yep.
Yeah. Apparently you have to buy this,
Because
but I'm
that's,
sure it's not
that's the
much.
first thing I thought of actually before the plaque assay was a colony counter. We used to have these things where you
Yeah.
stick
Yeah,
the Petri
yeah.
dish on the thing that's got the little pokey thing.
Yep.
Do people do that anymore?
Yes.
And you poke the colonies and it counts
Yeah,
them?
yeah, sure.
Far out.
Wow, this is very cool, Jason.
Yeah.
Thank you. Appreciate it.
Now, somebody with coding
And,
skills could probably do this with a webcam and a Python script or something.
Of course.
But
but
yeah,
not for
you've
us
got an
regular folk.
iPhone, and you might as well just buy
I'll
the
put
app.
my grandson on it.
There you go.
He's a burgeoning coder.
Wow, that's
Excellent.
really cool. Keep up the great, informative, interesting, and entertaining work. I gobble up each episode as soon as it's viral, Jason. Well, thank you. We really appreciate it. That's it for TWiV 873. You can find the show notes at microbe.tv/TWiV. If you would like to send in a question, a comment, or a pick, TWiV@microbe.tv. If you like what we do, consider supporting us. You can go over to microbe.tv/contribute. We are a 501c3. That means your contributions in the U.S. are tax deductible. Alan Dove is at alandove.com. Alan Dove on Twitter. Thanks,
Thank
Alan.
you. It's always a pleasure.
Rich Conda is an emeritus professor, University of Florida, Gainesville. He is currently in Austin, Texas. Thank you, Rich.
Sure thing. Always a good time.
And Brianne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.